Fine. So, hi everyone. Welcome again to Malaysian Research Insider. Welcome back if you've been here before, or welcome to those who are new. This science chat is co-organized by MBIOS, 100 Scientists of Malaysia, along with our media partner, Science Media Centre Malaysia. I am Sarah, and I'll be your host for today. And I am the co-founder of 100 SOM. So today we have Dr. Izzat Suffian, who is an assistant professor in drug delivery and nanomedicine at IIUM. His presentation today will be on his work in nanomedicines, a magical weapon to cure the world. Before I start that, I guess, Izzat, do you want the poll to be up? Or?
Yeah, that'll be great to see the poll.
Okay, we have a poll. Hang on, let me just pull it up. It should be up. Yes, do you think a pencil can save someone from cancer?
Okay, as I expected, people will answer no, a pencil can save someone from cancer. That interesting to see someone saying yes. So basically, from why I'm asking this, this question, because it will be related to what I'm going to present after this.
Okay, cool. So we had a fun poll. Izzat's going to address that question, why he asked that question. And it's over to you now. It's all yours.
Okay, thank you. So hello, everyone. Good evening in Malaysia. I think good morning in Europe. And also, I don't know, good evening in the other side of the world. I don't know how exactly the condition in other part. So anyway, thank you for joining the session today. So my name is Izzat Suffian, currently an academic from International Islamic University in Malaysia in the Faculty of Pharmacy, or we call here as Kuliah of Pharmacy. So the topic that I'm going to share with everyone, just a bit what I call it as nanomedicine, the magical weapon to cure the world. So basically, I will answer the questions about why I'm asking the poll about pencil and cancer after this. So currently, as I said, I'm from IIUM, also from a research group, what we call it NanoRG, associate member. So this is just introduce my university. So this is International Islamic University Malaysia. So this is the main campus. So basically, IIUM have 5 campuses. We have in Gombak, which in Kuala Lumpur. We have KL campus in Jalan Duta. We have Kuantan campus and Gambang campus, which is located in state of Pahang, which is located at east coast of Peninsula of Malaysia. And we have 1 campus in southern Malaysia in Pago, which is located in Johor State. So, and this one is my faculty called Kuliah Pharmacy, which I'm teaching all my pharmacy students, undergrad and postgraduate over there. So, this is, as you can see here, so this is actually one of the image that I did back in UK when I did my PhD. We imaged a lung cancer cells A549, which actually taking up what we call a material as from carbon. You can see the gold color here that actually try to penetrate or try to deliver to the cancer cells. So the green color is the cells, and the red one is the nucleus, and the gold one is actually the carbon. So now I'm going to answer the poll. So why I'm asking whether a pencil can actually save a cancer. So most of you, as I expected, will answer no. So you will expect how exactly one pencil can save the cancer. So I'm talking about the graphene that been used as the lead of the cancer. So graphene is type of carbon. So one type of material that actually nowadays people try to work on it to deliver drugs, and even try to use that to actually treat the cancer cells. So what exactly? So carbon nanotube is the gold color here, is part, or carbon needles is also part of the material that from carbon, and whereas it's graphene. So this is graphene. So the purple one is the graphene. So the colorful one in the middle is actually E. coli, or we call a type of bacteria that actually you can find easily when someone got infected with food poisoning. So this image was taken also when I did my PhD back in London by a mistake when I did an experiment. So basically we tried to observe the graphene, which is this purple color at the back. with a microscopic technique, which we call it as TEM, or transmission electronic microscopy. So we try to image that one, but during the preparation of the sample, without we realize we use a non-sterile water. So non-sterile water, which is a water that you not sterile them, that a possibility you got contamination from anything, including bacteria. So because we used that water by mistake, we got contamination from E. coli. But thanks to this bacteria, the graphene sheet attached to this bacteria that we can easily observe the graphene sheet. So this graphene actually that we use in pencil. So that's why we can use, not literally use pencil to save cancer. The material that made the pencil, the graphene inside, actually been used to deliver drugs, and also been used as to do some kind of a diagnostic or try to screening the location of the cancer cells. So what exactly I'm doing right now in IIUM? So this is basically what I'm doing, what we call a wet lab. I'm doing a laboratory work. Basically, we using many type of material. We try to working on many type of material. Try to use that material, make it as smaller as possible to actually deliver to the cancer cells. So now I'm going to just briefly explain what exactly is world of nanocarriers. So what actually NANOCAR is all about. So nanocarriers, easily to say, is a material that when we talk about in size, it's about 1 to 100, 200 nanometers in diameter. So you can imagine, like, we talk about nano size. So centimeter, millimeter, micrometer, then comes to nanometer, which if I want to translate is about 10 over power of -9. So that's very, very tiny. And these nanocarriers can be made up of many types of material. So as you can see on the slide right now, we have a polymeric nanoparticle. We are actually based on polymer. Usually we use a biodegradable because we want to inject them or to be applied inside the human body. So we don't want anything that can cause toxicity. And we have also a dendrimer. We also have that made by lipid or fat, which we can find a solid lipid nanoparticle, which is like it's a bit hard type of lipid. And we also have liposome, which is a bilayer of a lipid that makes into kind of a spherical. And actually, we can also use metal-based material to make nanocarriers. So for example, here, we are using iron oxide. So this iron oxide being modified, and because it got the magnetic properties, this material can also be used for MRI and also to localize the cancer by using a magnetic system. And this one that being used type of graphene or type of carbon. So here is a carbon nanotube. So it's the carbon sheet that we make into a tube, a cylinder. Then from there we kind of like decorate this cylinder with drugs and any targeting material that we want to target the cancer cells. And actually we also work with biological based material. For example, this is derived from virus, and this one is derived from protein. So we can modify this one, biologically modify them, and to make it into kind of a nanocarrier and so on. So we work with many types of materials. So as easy to say, nanocarriers is kind of like you have a car and you decide that car will only arrive at particular destination. So in this, to interpret into a nano world, so that car is a nanocarrier, and we modified that car to actually only arrive at particular cancer cells. For example, we want that car to only target breast cancer. We want that car or that nanocarriers to actually only target brain cancer and so on. So, and then that car, we can use many types of material to make that particular car. So this is all the material being used as nanocarriers. So what are the benefits of using nanocarriers? So because why we not using just a drugs? So as you can see in cancer therapy or like in therapeutic to cure the cancer, you know that the anti-cancer drugs have a lot of side effects, and this side effect can cause fatality to the patient. So what happened with this drug is actually really work to cure cancer. But what happened then if you give it just raw drugs? It will also kill your healthy cells. So we want to, as much as possible, reduce the side effect. That's why we need to have a particular carrier, which we use this nanocarrier. So why nano? Why not micro and so on? Because cancer cells are really tiny. as a microsite. So to actually allow a material to actually go inside the cancer cell, we must have something that much smaller than the cancer cell. So in this case, the approach is using a nano-sized material. So the advantages of using nanocarriers: first, it can alter drugs pharmacokinetic, which I explained earlier, which, for example, anti-cancer drug, it can go to the cancer cell, but at the same time, it can cause side effect to the healthy tissues. And also, sometimes when you inject it or you're giving orally to anti-cancer drug to the patient, not all the drugs actually arrive to the cancer cells. So in terms of like a percentage of drugs that actually can kill the cancer is really low. So by having that drugs inside a car and then only being active when it's arrived at destination, in this case is the cancer cells, it will help increase the percentage of drugs arrive at our target area, in this case the cancer cells, and allow the killing to be happened much quicker without higher side effects. And also, because we're talking about nano size, so it's small dimensions. So because it's really small, as I said earlier, it can easily penetrate to the cancer cells, because cancer cells are micron size, it's 3 times larger than the nanocarriers. So because of this small dimension, you can easily have that particular carrier to be penetrated and then release the drug inside. And then, because we're talking about nano size, so for example, in a 1 ml volume of drugs that contain these nanocells, these nanocarriers, you have a large surface area. Thanks to this large surface area, it allows for modification. For example, to have some kind of like a key that only can unlock the cancer cells. So in science term, we said it's targeting moiety or receptors that only can bind to particular cancer cells. And also with the large surface area, you can modify them to have drugs inside and outside, and so on. And number 4 of the advantages of these nanocarriers, because as I explained earlier, it made from various molecular structures. So from lipid base, polymer base, from metal base up to the biological base, this kind of material can work on it, and you can make the nanocarriers based on the purpose of the medication, either for therapy or either for diagnostic, and also depends on the location of the cancer cells. So it can be topical application, it can be systemic application, and so on. And thanks to these nanocarriers, some of them are empty inside, some of them also is a quite a defined surface. You can actually allow biomolecules or anything or any imaging props carrier. So with nanocarriers, it's not just can carry drugs. You can also carry fluorescence or fluorescein. You can also carry radioactive material that allow for the nanocarriers can be used not just as a therapeutic approach, but also can use as diagnostic approach. So the ideal of the current nanocarriers work, we try to use kind of approach what we call as theranostic, which actually a therapy plus diagnostic, meaning that one nanocarrier can work to kill cancer, but also can also use to trace the cancer cells. So that's exactly a general advantages of nanocarriers. Okay, this one I'm trying to explain number 5, how exactly you do the screening or diagnostic. So if you carry a fluorescence, so you can use a fluorescent image. So for example here, this in vivo imaging system, which I did all these 3 back when I did my PhD back in King's College London. So we have fluorescent approach where we have the fluorescein attached or conjugated with a nanomaterial that we can trace where exactly the cancer cells. For example, here the brighter the color, the high intensity. So we can see like our nanocarriers that carry the probe that have the fluorescein actually more accumulated in the tumor cells compared to the other part of the body. And then we have what we call nuclear imaging. So nuclear imaging, you have a radioactive material that conjugated or labeled to the nanomaterial, then you can where exactly the nanocarriers went to. So of course you want confirmation that our nanomaterial actually localized in cancer. So this is actually my paper where we try to localize the HER2 expressed cancer, which you can find a lot in breast cancer cell. And then actually the cancer, we have a model that in the nearby the around this area. So we can see that after 24 hours, all our material actually localized where the cancer located at. And then we have what got luminescence. So luminescence, easy to say, if you know certain sea life, like for example, squid, jellyfish, you can see sometimes they give kind of like a bright color at night or in the dark. So that color is actually luminescence. So with the luminescence, usually we use this approach to monitor our cancer model to know whether, for example, this we use the mice model. So we want to know the growth of the mice model, whether the size of the cancer is getting increased and so on. And then from there, we can do kind of localization with the nuclear and also the luminescence, whether actually our material is located in the cancer or not. Okay, I'm going to share a bit of my PhD work and then what my current work. So in my PhD work, I'm using hepatitis B virus core particle, or we call it a HBC. So it's the type of particle that mimic how exactly the virus work, but without carrying any DNA or RNA viruses. So because we know that virus is easily penetrate and infect our healthy cells, so we want that properties so that it's easily to infect the cancer cells. So we modify them so it will not carry any viral genome, so it will not be causing any immune response. Also, it will not cause any toxicity to the body. And also, we modified with some. We have kind of targeting moiety. So this nanomaterial, this HPC is hollow, so it's kind of ball, so inside is empty. And then in terms of size, it's really, really small. It's about 30 to 34 nanometer in diameter. And then actually you can see all this red color. So one of this red color is one monomer. So it made from from 180 to 240 units of monomer. And then because of the have a very high number of monomers, it gives a strong protective capsid shell. So meaning that when you have something inside of it, you put drugs or you put some kind of plasmid or art, any protein inside or any material you want to put inside, it will protect that material degraded before it reach the cancer that we are targeting. And also, thanks to the property of the HPC, we actually can open, or we call it as assemble or disassemble. So you can open and close. This nanomaterial easily by modify the salt concentration. So whenever you want to encapsulate drugs or any biomaterial or any material you want to encapsulate inside, I just increase the salt concentration, so it will kind of like open it a bit and then reduce back the salt concentration that you can close it, so you can encapsulate any material inside of it. So this is what we call it. We use kind of imaging. We have to use cryo-TEM. So this is the material, and then this all the bright colors. Actually, we use AFM or atomic force microscopy. It's another type of microscopic imaging system. And then we can also love of quite a homogeneous of the material. And with this HPC, I actually label them with radionuclide or radioactive material. So the radioactive material that we use here is, we call it 99M technetium. So 99M technetium, for those maybe from the medical or maybe a radiographer, is quite common in Malaysia. I think in other part of the world, we use that for diagnostic imaging. Why this one being chosen in clinical? Because whenever the patient being given technetium, it will excrete it after 24 hours. So it will not cause the patient to stay a long time in the hospital. So after done the diagnostic process, then you can allow the patient to discharge from the ward after 24 hours. So we use this one because we want to use this in clinical. And then the model that I use, so we call it interperitoneal model. So the cancer cells that has HER2, we allow it to actually grow inside around the stomach cavity. So and then we try to use this material whether we manage to deliver to the cancer cells or not. So with the imaging this one, so we give injection to the mouse. So this at the first 30 minutes, so you can see the material is all over the place, but it's already localized at the cavity around the stomach cavity. And after 4 hours, you can see in the bladder, and then these are actually the cancer cells. So after 24 hours, we realize that we observe our material actually only localized in where are the cancer located at. So it means it's really good our material, our nanocarriers, which is BC, manage to only localize in the cancer cells. We don't observe in other part of organ, even the lung, the heart, even the brain, or any joints. So meaning that with this material, with this approach, we can actually send drugs to target the cancer cells. So in future, when this being commercialized, we know that in terms of cancer therapy, we can prevent a lot of side effects that sometimes cause fatality, and then all the drugs only work to kill cancer because we just deliver the drugs to where are the cancer located at, rather than whatever we don't, we just inject the drug and it goes all over the place and try to kill other cells, especially the healthy tissues. Okay, that's my PhD work. So this is what I'm currently working in IIUM with my laboratory right now in my research group. So right now we try to approach for oral cancer, because in Malaysia, the numbers of people are smoking. Now we got vaping people are increasing, and then those being exposed with long-term smoking or vaping have high potential of getting oral cancer. So, and what happened in Malaysia, whenever these people that been diagnosed with oral cancer, they're usually at the late stage 3 or at early stage 4, which means that has quite a late stage of the cancer. Usually we divide cancer into 4 stages. So what happened then, the cancer already developed, because usually with oral cancer, you observe that the oral cancer is only kind of like an ulcer in the mouth cavity. So you thought it's just an ulcer, it's just having the typical ulcer treatment. But then after a while, when you do some biopsy, and then the doctor do some diagnosis and check, and realize already at oral cancer, but at late stage. So we want to use this drug, what we call it 5-fluorouracil 5-FU. So this drug is commonly used for digestive organ cancer. But the problem with this drug, we inject the drug directly, is actually non-selective distribution. So it will go all over the place. And then again, as I said earlier, it will cause toxicity to healthy tissues. And also it got short half-life. So whenever it goes inside the body, it's immediately been degraded, and then the efficacy of drug to treat cancer becomes lower. So our aim to use this drug and modify it using nanotechnology, actually encapsulate the drug, so that we can reduce what we call it as the half-life problem. Also, we can specifically deliver the drugs to the oral cancer cell. So this is the work I'll explain. I know it's very technical here. So right now we're still in the development process. We already managed to have the drug inside the polymer. So the polymer we use, we call it as PEG-PLGA. So this particular polymer is actually safe. It's already FDA approved, and then it's non-toxic. And whenever it goes inside the body, it will degrade it after some time. So it will not leave any traces inside the body. So it will not cause any immune response from our system. So we managed to have this particular material, this NANOCAR with this F5FU drug, and the size that we got about 200 nanometers and below. So we already have this particular nanomaterial. So right now, because right now in Malaysia, MCO is a bit put on hold on top of the research work. So after this, we will use this material to try to test whether it can give improvement in terms of like killing the oral cancer cells or not. So hopefully this work will have a nice result done by my student. So that's pretty much of my research work. So if any of you would like to know more about my work, or thinking of maybe we have a collaboration, you may contact me. So through my email, this is my official email, and through Facebook you can find Izzat Suffian, and to Instagram is Izzat_Suffian, and also on Twitter Izzat_5. So you can contact me any of this. I will happily to entertain you, and hopefully we might have a future collaboration afterwards. So this is my team. So if you can see here, I'm the only guy in my team. I don't know why. I try to get male students to my group, but all the girls come into my group. But it's okay. I love working with anyone. So I'm not like sexist, I'm not like feminist, but I'm welcome everyone to work with me. So anyway, thanks so much to IIUM for giving me the opportunity to work there, and my research group, and NanoRG, and then also member of CRS, Malaysian Local Chapter, and also official member of SES, the main ACS in the United States. So thanks so much, everyone. So if you have any questions, feel free to ask here. through the Zoom chat, or maybe, I don't know, maybe we have questions on the Facebook Live as well. So I give back the virtual floor to Sarah then. Thank you, Sarah.
Thanks, Izzat. That was a really good presentation. And I like how you make it so concise, but then like it really packs a punch. Like you got the point across, but really in a very compact manner. That was something that a lot of us could learn how to do so, especially in academia. Otherwise, you just sit in a 1 hour.
To be a lecturer, you need to like catch time to ensure that you can deliver the message.
Yeah, because otherwise your students just fall asleep. But that was a very good presentation. Okay, we have a few questions. Okay, so the first one, it's more like general nanocarriers kind of thing. So are there, for example, okay, I think we mentioned this before because Izzat was featured on 100 SOM before as well. So I interviewed Izzat, so we had a bit of conversation about this, I think. But are there any considerations for the materials that you choose to use in nanocarriers for different communities? For example, in Malaysia, there will be a more halal kind of side to things. Are there considerations that have to go into that?
Okay, yeah, I think for the Malaysia or perhaps the Muslim community, the approach also we want the halal material. So that's kind of like we to ensure that the material, when regardless any religion, so our target right now, like regardless any religion, any special diet, for example, we have people that are really vegan, so you cannot have any animal based. So we try to use that approach, vegan and halal, so that it can be reached and be used by any community. And one more thing is like a lot of material being used in the nano are quite expensive at the beginning because the demand is very low. So the new approach being done right now by a lot of researchers in nanomedicine, they try to use a plant-based material. So for example, in my group right now, we try to work with hydrogel, which actually in other words is agar that used for it. So we use that material instead of just using for causing you might diabetes, it off a lot of sweets. So we use that material now for cure. So we try to use a different approach, not just expensive approach, but also the cheaper approach, so that the therapy will be much cheaper in the future. Because I know cancer, I mean chemotherapy causing like 20,000, 30,000. That's the cheapest I heard.
Yeah, and then if you add on something more expensive to that, who can really afford it in the end? So
yeah, who can afford it. So we need to ensure that people who have like a less access to the medical can access this easily, rather than they feel like they need to sell their kidney to have a chemotherapy.
I understand. Okay, thanks. Next question. What is stopping the use of nanocarriers for treatment?
What is stopping the use of nanocarriers for treatment? Okay. What exactly that we have tried to find is a bit the challenge of us, like the immune response. So some of the material are quite strange. It's not being used by our body. So we need to ensure that when we're giving certain nanocarrier for certain material, that the body will not recognize as come alien, and then it will cause immune response. That will cause another trouble. So certain material that we know that will cause immune response, we do some different coating. Sometimes people use sugar coating to ensure that our white blood cell will not recognize this as kind of like harmful, so that it will easily do the treatment. So that's one of the challenge when using nanomaterial here.
Just curious then, are there any long-term effects from like nanocarriers, especially if they're not usually put into the human body, right? Even if you change it around.
Yeah, I mean, that's really great question, because the nanomaterial studies is really very short. I should say it's less than 50 years. So people are not really look into the long-term effect in terms of the health. Now we have a data that talk about environmental. I mean, that not used in the medical, but for example, we can find a lot of nanomaterial now being used in cosmetic. So people are trying concerned what will happen to the with the environment. So a lot of data being coming about the environmental effect and so on, the quality of the soil and so on. But with the health point of view, we still lack of data right now. But I think that might be a good study if someone wants to look into it, the networking and the data of whatever the cost in long term, because we have no much data about it yet.
Okay, interesting. What's the longest study then, like longitudinal study on the long-term effects of nanocarriers?
So the long-term effect that people want to know, like especially like the non-biodegradable one, like if you use, like whether if it stay in the body, what will cause in terms of that the long-term in terms of the health issues. Maybe it will affect our neural system, or going to cause a mutation into our DNA, RNA, and so on. So that should be looked into, because like even though the approach we tried biodegradable, or we expect the material will be excreted naturally by our body system, there's no 100% ensure, for example, the metal base, it will excrete it after a while. We cannot say 100% because sometimes you lose of detection. So those are kind of challenge as well.
Okay. Okay, next question. I think this is a bit more specific on your research studies, I think. Which nanocarrier is better for MAB, like PD1 or and RNA? Could you maybe take some, make some time, what's MAB,
so
PD1?
MAB is abbreviation of monoclonal antibody. So basically, like, the question is, I know this person, Lukman, is my friend. so basically, like, NANOCARE is better. Which NANOCARE is better to deliver antibody? So monoclonal antibody usually have quite a large size for that, and then to ensure that what we do to send monoclonal antibody, we modify the antibody first. So we only have the active part of antibody to actually work on it, and then to ensure the size is suitable. But in terms of material, at the moment, I can see all the material that I introduced in the presentation, a lot of them have been using antibody approach. Either the antibody be used to target cancer cells, or the antibody itself become the treatment. So if you're thinking of the antibody to become the treatment, perhaps we can encapsulate the antibody inside, but usually we put the antibody kind of outside of the material so it can catch whatever the receptors outside there.
Okay.
Yeah.
Hopefully your friend is satisfied with your answer.
Yeah, I think he will text me after this to ask more questions. Yeah.
Okay, next one. So someone, Leihian, says, congrats for the 5FU encapsulated with your PEG polymer. What is your plan for increasing specificity of the nanocarrier to the oral cancer cells?
Okay, for the specificity here, like the oral cancer that I'm working right now, we have a receptor, what we call it EGFR. So it's kind of like, so each cancer have their own ID. So they have their own ID. So one of the ID here, I would call it or receptor in the oral cancer is EGFR. So for my formulation to actually go with this one, I need to have something on my nanocarriers to only attach to EGFR. So meaning that it only recognize specific EGFR expressed oral cancer. So then we can increase specificity. If you don't have this kind of targeting moieties, what will happen here then, your material might go other type of cancer cells that you not intend to go, because like it's still small. The possibility to be engulfed or being captured by other cancer cells is still there. It's not high, it's still there. But then we want to specific goes that you want most of the nanomaterials, in this case the nanocapsules, goes to our cancer cells. We have targeting moiety that's specific to the EGFR here. Okay.
So how does your nanocarriers react against the oral cancer? Would there be any immunosuppression activity or pro-inflammatory mechanism that will affect your drug efficacy?
Okay, so usually when we do what study with the nanocarriers, so usually what we do, we try to study the immune response system first. So with the immune response system first, we want to know like whether our body system will react to it, because if it reacts to it, all the nanocarriers will be excreted by our immune system. So here, with certain material, we realize it will not cause immune response. For this case, I use PLGA, PET PLGA, it being approved by the FDA. It will not cause, it will cause a bit, but not significant immune response. So, and then the pack, if you see, I said PLGA pack. So the PEG is the material that can escape from. So usually with any smaller material goes inside our body, it goes immediately into our liver. So our liver is kind of a huge filter. So with the PEG polymer, it will escape that liver. So it will keep circulating into our body, then as to allow the exposure to the cancer become longer. So here we can ensure that nanocarriers will reach to the cancer cells. So usually when it reaches the cancer cells, it will penetrate inside by collateral mediated if it's protein based, but because it's a polymer based, it will engulf through the channel of the system of the cancer cell. Sorry.
Okay, so if someone has like depending on the comorbidity of like the diseases that they have, will any like immunosuppression or like inflammation kind of mechanism that they already have that will decrease the drug efficacy or affect the drug efficacy?
So it will not say the drug efficacy, it will might affect in terms of the distribution of the nanocarriers. So if the nanocarriers somehow, because of this particular disease, causing block the nanocarriers to arrive at the cancer cells, then automatically the drugs is not reaching there, right? So, but yeah,
Okay.
that's we never try to really goes with that different of inflammatory diseases, but this is a really good idea. I think not many studies try, for example, the proof is working. How about like in different cases of people with different immune response system and mental disease, whether it gives a different result or not?
Yeah, that's true. Yeah, if someone like say did a organ transplant or something, then they're on immunosuppressants, but they have cancer and need treatment. Like, what's the interaction between those? Because both is a matter of life and death. So like, how do you
that's true.
balance?
So that's usually like people that we try to ensure that how much of the immune response will be given by the patient. Like if one of it is higher, we'll try to different approach them. So in this case, NANOCARE is being used for mainly for chemotherapy drugs, and now we also use the radiotherapy, which exactly the NANOCARE is just without any drugs. It will go to the cancer cells, but then with the radiotherapy, it will release radical that actually try to kill the cancer cells.
Okay. What are the limitations of your study? Can there be a delay mechanism built into the carrier?
Okay, the delay that usually people actually facing, like you got a nice result in vitro, you kill the cancer. When it goes to in vivo, it gives you the opposite. The animal, that's in animal. And then another stage is that it works until in vivo, and when you go preclinical, it's another story. So that's usually happen when people in drug world, drug discovery, drug delivery, everything's so nice in vitro and okay, nice in vivo, test to the clinical, and then it's rejected. So this is quite a challenge. And then of course, another thing we talk about material, we talk about cost. Because some of the material are really expensive, and then we need to talk about like, so with the research nowadays, we cannot just talk about in terms of the lab level. So it should including you want to be commercialized, not because you want to make money. Money is another story, but you want to ensure that it can be used. I mean, in the pharmacy, in the hospital. So you might thought your method in the laboratory working fine, but you want to scale up them. It costs like per treatment, perhaps now 20,000, maybe 10 times more. So that's another challenge with nanocarriers. You need to prepare a method. Then when you're scaling up, it gives you a really low cost, but a high efficacy and efficiency.
Yeah. Interesting. Yeah, that's definitely something to consider. Can you build a delay mechanism into your carrier?
Yeah, we can build a delay mechanism. Like for example, with the polymer base, you can have a multi-layer of a polymer base. For example, like you can do kind of modification to slow release of the drug, or perhaps like a multi-stages of the drug. Perhaps you have two, three types of drug inside. So it will release bit by bit based on the type of material. So now we're not just working one with nanocarriers. Perhaps what I experienced, we use outside is polymer. Inside I have another two types of nanocarriers, where exactly one carries drug, another one actually a metal nanocarriers, because you want to use for the MRI purpose. So now we can have like a multiple type of nanocarriers based on that. You can delay them, you have multiple release of drugs, and so on.
The techniques are really getting more sophisticated as time passes. It's just getting smarter.
Before that, it's just a one material, one drug.
And now it's just like a cocktail of
a cocktail of many things. And now we try a new approach. Like now people are talking like, can you make that nanomaterials self-intelligence AI? So they try to like, how about we put some chips so then the material
they know
can just
what to do, when to do.
Swimming around inside the body, we know what to do. I said, okay, that's another story. Now you might see everything in Black Mirror in Netflix might become reality.
Highly possible though, that 30 years down the line when our generation have retired, God knows what they will be creating by that point. So,
people never thought about nanomaterial can be created 50 years ago.
So things
people
are
not just the drug, it's just like in
changing.
capsule, in tablet.
It's constantly changing. Okay, this one goes a bit further out, zooming out again. So I think about the materials that you use to make your nanocarriers. does tocopherol, natural vitamin E,
Okay.
makes drug delivery more efficient?
Okay. I really like that question because it's very specific. I don't know who's asking
Yeah,
this because like
it was from Instagram. Yeah.
I'm working with vitamin E right now.
Oh.
So I'm working with my students. So basically, vitamin E, we have two types, tocopherol and tocopherols.
Mm,
So the vitamin E has been studied by one of the professor in USM,
mm,
mentioned that it got cancer therapeutic properties.
mm.
So vitamin E can help into cure cancer. But then, okay, please do not take this statement literally. So after this, people might buy vitamin E in the
It's-
pharmacies. Okay, don't get. I take and make sure I have disclaimer here. The way it must deliver, because when you consume oral, you don't know how much percentage. So,
mm,
but the one to be studied with the, I forgot his name because I met him a couple of times. So he directly apply the vitamin E to brain cancer and also to Alzheimer, if I'm mistaken, and you can see some kind of improvement. So with I'm working right now, like to see if we deliver directly this vitamin E. So I'm not going just because vitamin E out there is not a sole tocotrienol or tocopherol only. It's a mixture. So I'm going to just solely choose one of these to see differences and try to see in any type of cancer which cancer actually can be treated with this tocopherol, tocotrienol of vitamin E. So a lot of study. I think this person who asked me is really studying about vitamin E. Okay, please do not make a business vitamin E can cure cancer after this, okay?
Yeah, don't make a statement.
But
Vitamin E cancer cures, and then start
yeah, it got
viral
potential.
WhatsApp.
Yeah. After this, people will put my face and say, Dr. Izzat from
Yeah,
IIUM saying that vitamin E can cure
yeah,
cancer. Oh
yeah.
my God.
You go famous, is that? But for the wrong
In a
reasons.
bad way, I think.
Okay, yeah, yeah. I think scientists need to make sure, like, when they say things, they don't. They're so used to saying it to an academic community where people are like, "Oh,
Yeah.
okay, I will be wary," but the general public is it will just take you literally for word. Like,
They will cut
yeah,
that quote and
yeah,
then put it
yeah,
big.
yeah. Copy and paste across your face.
Yeah.
Um, okay. Beyond cancer, how successful is nanomedicine to deliver drugs to cells in other diseases such as cardiovascular, heart diseases?
Okay, so basically nanocarriers, the really what's a magical, you can modify them based on what disease you want to target. So we
Mm,
also use nanocarriers for cosmetic. I think for those knows like they now have micellar water and everything, that's actually nano approach. And
mm, mm,
also we use for wound healing because like the penetration to the skin is much quicker with the
mm,
nano approach. And then with other disease, now we try to deliver insulin, synthetic
mm,
insulin, because it's much quicker rather than because insulin is very large molecule. So you make it smaller, so it will quick be released and to help producing diabetic disease. And then with the cardiovascular disease, like if you know certain tissues, they have a certain biomarkers, so you can actually specific to deliver that nanocarriers, perhaps certain drugs, perhaps, for example, you got heart attack, like immediately target to particular that tissues, the muscle tissues at the heart, and then it will immediately release the tension of the muscles.
mm.
So with
Okay.
nanomaterial, you can modify them, kind of like you have a universal thing. Whatever you want to do, you can modify them.
Yeah, yeah,
So
that is true.
that's why it's a magical. So of
Mm.
course you can use that for war. Okay, that's if you look at the title, to cure the world. Okay, I'm not talking about like the war part, but in terms of disease. Now we have
Yeah.
COVID-19 with the virus as well. So I'm also trying currently working with COVID-19 to actually for the screening using a nanomaterial approach.
Yeah, I think we have a question on that, but I'll finish Jake's question
Okay.
first. So the second part to his question was, is it easy to reuse carriers with different drugs for treating other diseases, or do you need to like revamp the whole thing completely?
So it depends on what some of the material can use the same material, just you change the drugs. Some of them that you can use similar drug but change the material. So I cannot say it's a linear answer or a direct one-to-one answer, but then it depends on
Mm,
how
mm,
exactly. For example, for cancer, usually we go specific because like we want this specific cancer, but then if you have a general formulation, okay, now for example, I have my nanocapsule, PLG nanocapsule.
mm,
So if I want like to have a target certain other type of disease, then I just modify the surface of it and then put
mm,
inside a drugs that I want to particular. So it's not that really hard, but then it depends on your approach actually.
okay. So coming back to that vaccine thing, so someone asked on Instagram as well, I think, about using siRNA for vaccine. So if you could just, so siRNA is small interfering RNA, which is like non-coding RNA molecule.
Yes.
So is that considered a nanocarrier as well, or like?
So sRNA is not nanocarrier. sRNA is as I said, is small interference ribonucleic acid.
Yeah.
So actually, there is a non-coding, but we use that to actually knock down certain genes. So that's my PhD project, gene therapy. Okay, so sRNA being been known by a lot of researchers, like it can knock down genes. So for example, like if you know this particular gene causing you to have a potential to have breast cancer, so you knock down that gene, then you will not have high risk of breast cancer, or perhaps you have a colon cancer. So you know this gene actually functional to causing that colon cancer grow very fast. So if you knock that gene, you will stop the growth of the cells. But with sRNA, you cannot directly inject to your body. It will immediately degrade it by your plasma in the blood. Got a lot of enzyme there. So what we are doing to protect the sRNA to be delivered, you need nanocarrier. And in my case, I use a HPC, but a lot of people using other type of chitosan, people use liposome, so it depends. But sRNA is not nanocarrier. It's a
something
nucleic acid material.
Yeah.
It's nucleic acid material that we use for gene therapy usually right now.
Okay. Right, I think we have one more technical, general technical questions before we close it off. You mentioned that targeting nanoparticles to oral cancer is by binding of EGFR receptor, epidermal growth receptor. Yeah. Does this mean the delivery can be done in any tumor types as long as the receptor to target is known?
Yeah, theoretically, that's what the nanomedicine approach. You know the biomarker or the target of the cancer cells, but not just it got that marker, it must overexpress. So I mean like it's intensity. So a lot of certain, because one cancer doesn't have one marker, have many types of marker, but you want that a specific marker that distinct for that particular cancer. So you know that marker, and then you have a targeting molecule that specific target that marker. So theoretically, it will only deliver to that or target to that particular cancer. Yeah, as what he said is yes, yeah, you can do this.
Okay, yeah, I think that was the last question. I mean, we had another question asking about how do you direct the nanocarriers to the cancer cell, but I think you've addressed that a few times with the other questions. So yes, okay, I think that is it for the questions. I think I personally have a question for you. I think I'm just curious about where you're heading. Like, are you going to be working on nanocarriers for the rest of your PI career, the rest of your life until you retire? Just nanocarriers,
It's like I'm looking into like, because like especially in Malaysia, people are not really work much with nanocarriers, especially in nanomedicine. In terms of like semiconductor or this engineering part, we got a lot of people working on environmental. But in terms of nanomedicine, people are not really work on this because they know it's hard. You need a lot of patience. You cannot have like immediate results. And for me, like it's a thing that I can contribute to the country.
Yeah.
Because like in Europe, a lot of people working with nanomedicine in the US, and when I did presentation in US, the presentation in the other part of the country, like they're quite surprised. Oh, we have someone from Southeast Asia that working that's really something very tedious like this. Because they know the approach, and in Malaysia, even the technology, I say we are still have certain limitation compared to the other part of the world. So if I want to say like 5 years, 10 years from now, I would love to still work because I want to see at least one of my formulation will be used in the hospital.
Yeah,
because I don't want like I kind of like what we call it short sendiri, like doing all the lab work, but then just in the lab.
Yeah, it doesn't make sense to stay in the lab. Yeah.
Sometimes the people just for the sake of publication, for the sake of have more papers, but that's it. But for me, like my purpose to solve the problem. So a researcher should or a scientist should have the mindset why you are in this kind of field, because they are probably want to solve. So until the problem been solved, you should work hard on that. So if you have that kind of connection, whatever hurdle you are facing,
it'll push you.
You will push you. Because for me, like every time like I do nanomaterial, even though like you cannot see with your bare eyes, I've got a nice result. I can observe under microscope. Like I'm like kind of very high. Like, oh my god, I got something.
Yeah, that was what I was going to ask you as well. Like, how is it like working with NanoCare? So obviously you can't actually see what you're working on, right? It's just like, just pipetting a lot of small stuff and then looking under the microscope and then saying, oh, I think it worked. Like it's so hard to deal with that kind of.
Yeah, that's true. Like especially like you did formulation, you see in the vial, oh, it looks nice, but then the anxiety always come when you want to measure it. So actually, after I done one formulation, we have a machine called synthesizer, or then we have the nanosizer that actually measure all the material in nanosize. So whenever I put that material in that machine, press the start, I was so like anxiety, like the anxious, like please, a nice result, only one take, only what the size I want. Like that's always kind of feeling like after like I've been working like from 2011, now it's 2020, like nearly 10 years in this nanomedicine world. Like still the anxiety is still there. Even like we said, you already at the academy, you already got PhD. No, the anxiety is still there. Like I'm working with new material, new formulation. I was like, oh my god, please, like this kind of feelings coming.
Yeah, but it's good. Like I mean, at least that means that kind of shows you're still passionate about research, isn't it? If you are like, oh, you know, I really couldn't care less. It works. It doesn't work. Okay, lah. Then it's just like
that's kind of people just like for the sake of publication.
That's dangerous now.
You do research for the sake of you want to get, for example, just want to have title doctor of PhD, or you do the research because my company asked me to do, or you do research because I just want to being promoted to associate professor or professor. If that's your target, your passion to the research will end immediately afterwards.
It's not hard.
But if you put your mind such, I want to solve the problem up there. So every time until the problem is solved, you keep passionate to do the work.
I agree. Oh, it's nice to have like Southeast Asian representation. Go Izzat, like keep going. Have you had your PhD students graduate? Like, are they any of them intending to go into academia?
One of my master student will be graduating next year, and I have few more students coming this year as well. Supposed to be this year because of the MCO, everything like and then I think I mentioned in the 100 scientists from Malaysia post, like the problem in Malaysia, delay of getting items, all the bureaucracy thingy and so on. That also affect your research and also sometimes affect your spirit during research. Sometimes I just like, okay, ignore all those things happen, focus on my work. Whatever people think, I just do my work.
It's nice to see people who are so invested in their work, like actually wants to make a difference, and like Southeast Asia representation. I think the longer you spend abroad, the more you want your country people, or like Asian people at least, to come out and have a limelight. Like it's so dominated by Western culture. You just like there are great research in Asia as well. It's just people just don't see it, or it's just but that's my own personal view. But anyway, I think we've come to the end. Yes.
Yeah, like when I did my presentation last month, it's a virtual US, it's a controlled release society. It's really like people are working with the drugs. So in the group, like I think I'm the only one from Asia, Southeast Asia, and then Asian. Others are Western or Asia Pacific, I mean Australia or New Zealand. I was like the one Asian in that particular presentation. Like, okay, where are the other people?
How did you feel?
I feel like free, kind of like happy. Of course, I heard like, oh my god, I presented Southeast Asia, but at the same time, like I'm waiting all my friends that I never met as well. Like, please, you guys should be there as well. Like, show them we actually, regardless you're Western or Eastern people, actually, as long as you push yourself, you can at the same power with them.
As long as you're doing proper science, sound science, it all counts. You just need to get out there.
Yeah, especially I'm waiting for a lot of Malaysian to be there, because like to see Singaporean is quite common.
Yeah,
and I want to see more Malaysian over there. Like I know Singapore, I respect them with all the technologies, but Malaysia have the potential. Just like we need to push a bit more.
We just need to step out of our comfort zone. I think this is the one thing we keep hearing in 100 SOM stories as well. Whenever I ask them, so like, what made you successful? Like, what made your journey? Everyone's just like, you just have to step out of your comfort zone. So to everyone listening, if there is that doubt, like a voice of doubt in the back of your mind, saying like, I'm not sure if you can do it. If you're scared of something, that means you really want it enough as well. That was one advice I got. So just do it, guys. Yes.
Yeah, just do it. Ignore what people think. As long as you know what you're doing for the sake of others, to help people. To help people, not for the sake of you want kind of like people appreciate you. No, you want to do something to help the world.
I agree.
Okay,
with that inspiring end, to our session. I think that was a very good session. It was nice catching up with you again. Hopefully you're
Yeah,
doing
it was
well.
like a long time. Well, like I don't know, is it last year we checked? It's
Yeah,
last year,
yeah. I
right?
think it was December 2019. It's been what,
Yeah,
7 months?
almost a year.
Yeah,
Almost
I
a
know.
year.
I know. I like all your Facebook posts and your Instagram posts and everything, but it's not. I haven't actually spoken to you for a while. But yes, thank you so much for your time. Thank you everyone else for tuning in. And yes, for any more updates regarding our future Research Insider series, like our Facebook page, MBIOS and 100 SOM. And we will see you next Saturday.
Okay, thank you,
Yeah,
Sarah.
thank you. Okay, thanks everyone. Enjoy the rest of your night and or afternoon. Thank
Yep.
you.
Okay.
Bye.
Bye bye.